Okay, hi everyone. I'm Nick Mancuso. I'm super excited to be here today. So if you're looking at the title and you're wondering what happened to the fun and profit, I promise it's still buried in here. But I did a last minute pivot because the contents aligned very much with an earlier wonderful talk that David gave. And so I thought we would just maybe pivot less from the nuts and bolts of JAX and more to a direct application of JAX in some software that we've worked on recently. And so what I'll talk about is our model for calling eQTL data and eQTLs in single cell data. So obviously I don't need to belabor the point on this slide. I think we're all very familiar with what a GWAS is, what these points represent, and the fact that despite its massive success in identifying regions in the genome that track with disease risk or some continuous trait, we really only have a handful of examples where we really understand sort of mechanistically what might be going on in terms of how variants impact some nearby gene in some context that confers risk to individuals. And so, there's a huge wealth of evidence, going back a decade plus now, that the non-coding variants that are prioritized by GWAS are massively enriched for regulatory function and biochemical signatures that track with regulation in some way. And so the thought was that these risk variants are just dysregulating some nearby gene in some tissue and some cell type at some particular point in time, right? And so that leaves like a lot of conditionals to work our way through. But initially, one of the largest, like a phenomenal effort to sort of shed light on this was the GTEx project, which produced bulk RNA-seq in a large number of tissues in a massive cohort, in which individuals then identified genetic variation that tracks with changes in gene expression. And so this provides, so not only do we learn about how genetics may regulate nearby genes in some tissue of context, but by integrating those findings with GWAS, right, it provides sort of like this statistical foundation for prioritizing potential genes and having some understanding what these variants do. Despite that, the success of that, I think we should temper our expectations a bit here, because integrating sort of findings from bulk eQTL data with GWAS risk loci shows only a modest overlap, right? So 43%, if you're just doing like a strict co-localization or like significant in this and significant in that. While this is still great, right? Like this is a completely different data set and different people who may or may not have these diseases, and this still provides a lot of information to understand what might be going on. But still, if we do a stricter maybe analysis, or more careful analysis, I should say, like the findings from these types of tissues or these settings only explain 11% of the heritability of GWAS, right? So that's very, very low. It's saying that of the heritability we can identify in GWAS, the bulk of it is just missed by bulk expression. I didn't mean to do that, but sorry. Okay, and so really then, we were missing the context, right? Like in a general sense, whether it's the cell type or some time point. And so, one of the things that we also have a lot of evidence for is that gene expression regulatory elements are often cell type specific, right? And so what we have here are single cell attack-seq measuring accessibility across the genome and various blood cell types. And then what we see here is an eQTL that falls really in this regulatory element that's only observed in monocytes, right? And so it points this picture that this variant is really only acting in this cell type for this gene here, right? And so we need to, despite being able to just take a blood sample from people and do a bulk eQTL analysis, this would largely be missed if we were trying to do that. Likely be missed due to probably attenuation in the other cell types. And so this drives a need then to generate single cell expression data coupled with genetics, so we can quantify the extent to which common genetic variation tracks with gene expression at the cell type level. And so currently there are two data sets that are, I should say, the two largest data sets that are out there are the 1K cohort and the clues cohort, which came out around the same time in 2022, I think actually maybe the same issue. And so both of these have provided a phenomenal reference for methods developers such as myself, but also folks who want to understand how, again, disease risk might be mediated at the cell type level. And so, so to take a step back and say, well, what approaches can we use to identify single-cell eQTLs? Well, certainly we can just take sort of the off-the-shelf eQTL calling from bulk, apply some transformations to our data, and just treat it as if it was bulk expression, right? And then run it through. And certainly, like that's completely fine, right? That's a sound way to go about it. But one of the issues with single-cell data is that the counts are going to be much more sparse compared to the bulk setting. And so in the bulk setting, applying these transformations might not lose that much information under that transformation. And so it certainly won't be an optimal approach in the sparser setting of single cell data. And so that really motivates trying to model naturally just the observed counts rather than any sort of transformation on those counts. And I'm by no means the first person to say, hey, that it's count data, that's what we should be modeling it as. Differential gene expression analysis tools have been around for a long time, have used sort of natural count-based likelihoods to do association testing. But in eQTLs, you're going to have thousands of tests for one gene and one cell type, right? And so now you have thousands of genes, you have like maybe a dozen or so cell types, and so it's just not really something that's tractable. On the counter to that is a really flexible model, cis-regulatory, which sought to tackle this problem directly by saying we're going to model the counts we're going to model gene expression under a count-based likelihood while accounting for within individual variation. And so it's a very natural way, if you're familiar with these mixed effect models, to tackle the problem. However, I think sort of a first sort of pass on single cell eQTL data is that if your covariates are fixed across the cells, then a lot of the math kind of simplifies, and we'll see in a minute. There are plenty of reasons why you would not want covariates fixed across cells. And so I want to be clear in that this is a much more general model. But certainly if we want to just kind of a first pass, genotype is fixed across samples, it's not going to change across the cell, your covariates are fixed. Then what we propose is something we call JAX-QTL. So JAX is still there, right? We propose to use a negative binomial model. And we implement it in JAX because it allows us to leverage just with the, as David alluded to with like PyTorch earlier, CPUs, GPUs, and TPUs with just, you just add a flag. There's no additional compilation that the users have to deal with. There's no crazy code from our end that we have to do for specific architectures. You just write Python code, and at runtime, it'll do the right thing, which is really fantastic. And so, what is the model that we use? Well, it's really just a negative binomial model. You have a log link. It's a linear map under that transformation. And so this is fairly standard, right? And so we can fit like a null model under our covariates and accounting for sort of library size and things like that, and then perform a score test one SNP at a time at the locus. All right, and so for folks who are a bit familiar with these models, the reason why we didn't use a Poisson distribution is that it can be restrictive in terms of the mean variance relationship. And so oftentimes in gene expression, we see that not necessarily holding. And so the negative binomial provides a little bit of flexibility, and that we also have this quadratic term here. And so just one thing I want to highlight is that, so under our model, really what we're doing is, if you know the cell type, right? If someone says, okay, we've given you a label for all the cells, within a cell type, you collapse down to a pseudobulk, right, for an individual. It just means you add up their counts across their cells of a particular type, right? As I said, this is our variance for the observation. It's this quadratic. But if you do that same transformation under the Poisson mixed effect model, you get something that looks like this, right? And so it's actually essentially the same structural form, right? It's still this quadratic relationship. You have to do some transformation for the within individual variation, but ultimately you just get this quadratic variance function. And so one can show that, under either model, these things map, the alpha under this model or this x minus 1 under the other model map very, very closely. And so, there can be a benefit of modeling the particular specific cell counts if you want to account for, again, that within cell covariates. But if things collapse down, there's really just no added benefit. And it's just going to be a more painful on the cloud cost to look at. So maybe I should speed things up a bit. So we did simulations starting from the CGSI model, right? So we simulated data at the individual cell level and then collapsed down to do pseudobulk and applied our implementation of a negative binomial, our implementation of a linear Poisson, and then compared it to CGSI and TensorQTL. And so under the null, we found that all models and software were unbiased under the null with the exception of Poisson, right? And it's just that, again, if there's, you add these things up and the mean variance relationship breaks down, and so you're going to have a lot of false positives. Under the alternative, we see essentially identical power for the negative binomial under pseudobulk versus running CGSI at the per cell level to identify eQTLs. And again, in this particular setting, right, where the covariates don't change across cell types, we know the cell types, things like that, we see identical power. And again, it comes back to that, the sufficient statistics really just collapsing down to that pseudobulk level anyways. Great, so we did a bunch more simulations, that sort of convince ourselves that what we're doing is fine. And so then we went to apply it to the 1K data, where we have 14 cell types from whole blood, and looked at roughly 32,000 genes. We did a bit of filtering to focus on 16,000 genes that had expression at least 1% in the population, and then focus on common genetic variation. And so we compared our tool, JAX-QTL, using two ways to perform multiple testing correction at the gene level. One is just we do permutations within each gene, and then across genes just do some FDR. Alternatively, within the gene, instead of doing permutations, you can run this ACAT-V test, which is this very, very fast approach. And so that maps, this ACAT-V kind of maps what CGSI is doing. But the big picture here is that, essentially JAX-QTL and CGSI find similar number of E genes across the board. We outperform the linear model, especially as data gets more and more sparse. And this is due to the fact that when cell types get more and more rare, you have fewer and fewer counts, and that log transform is really just going to potentially, you'll take up a power hit. Okay. So great, we have a bigger number. So, to convince ourselves that this made sense, we then sought to see what was the replication rate in the clues data set. And we found, again, breaking things down across the cell types, we found fairly reasonable replication rates. We had lower replication rates when we looked in the Asian participants in the clues data set. We're attributing that to power because that's much fewer individuals compared to the European ancestry. But this is also contingent on this binary choice, right? It was significant in our data set, significant in their data set. And we can do a better job by looking just at the effect sizes, regardless of significance in the follow-up validation or replication data set. And so again, here what I'm showing are the effect estimates from the negative binomial in 1K, and we're comparing it to the effect estimates at the same SNP and cell type gene matching and the clues. And again, we see this very striking relationship. And so for folks who've read, familiar with the 1K paper, there's a figure pretty much exactly, we just straight up, replicated that figure, but using our approach. So while they found very consistent linear effects in some of the more frequent cell types, again here under this negative binomial model, again, that's what we're seeing. And so again, I think this demonstrates that the model is doing a great job at identifying eQTLs. And so one thing to touch on too is we wanted to see, okay, is our implementation faster? So I'm talking about JAX. One thing that's neat about JAX is, again, like I said, it'll do just-in-time compilation on the fly to match the particular architecture that it's running on. And so here, I know there's a lot going on, so I'll try to break this down. These are 3 cell types that represent essentially common cell type, maybe fairly common-ish cell type and a rarer cell type within this data set, right? To capture, again, maybe like some notion of sparsity in those three settings. And so if it's a dashed line, it uses ACAT-V. If it's solid, it's permutation testing. And so we see that in this setting where we have a lot of cells, that cis-regulatory tends to be the slowest across sample sizes. And so, sorry, I should also clarify, we did downsampling. So this is the 50 randomly sampled genes, and we downsampled at various sample sizes to get a sense of runtime across as n changes. And so we see that using a CPO with permutations, we still outperform cis-regulatory. If we run ACAT-V on the CPU, we're about as fast as just doing the linear model with permutations, right? So it's very, very fast. So if you're willing to take a little bit of a hit to power, you can just do ACAT-V and you're running, nearly instantly as if you were just doing a linear model. Similarly, if we switch to using permutations, we can use a GPU or a TPU. Again, we get a massive performance boost. I don't want the takeaway here to be that somehow our model using a GPU or TPU is magically constant, like independent of sample size. What's going on here is that we're not getting full utilization, right? Like this is just too small of a test for these things to really ramp up. And I would imagine that as n gets bigger and bigger, we'll start to see like these creep up. And so that's actually exactly what we did. So we did upsampling to mimic the 10K that should be out hopefully soon. And so here, if you run cis-regulatory using a GPU with permutations, we're about 1,500 times faster than cis-regulatory. And if you drop the permutations and do ACAT-V, we're 10,000 times faster. Right? So that means when the 10K data set comes out, if you want to analyze this data, it's going to be very fast. Right? So I think we're all probably tired of just numbers, more, faster, slower. We wanted to come back to biology, and so then what we did was integrate a bunch of functional annotations. And so our hypothesis initially was that promoters are going to be much more shared across cell types compared to enhancers. And so by looking at the cell type specific level, we should see a greater enrichment of enhancer annotations compared to what we would see if we had used bulk. And so what I'm showing here in black is if just like we took all the reads across all the ignored cell type and just did, just completely drop that label and just treat it as bulk compared to single cell eQTL. And then what we do see is a greater amount of enrichment for cell enhancers at the single cell level compared to bulk eQTLs. It's not a significant difference because of the standard errors, but the hope being is that as data sets get larger, as we have more findings, this is something that will get a bit more fine-tuned. Those were using just like kind of broad regulatory annotations and not things that are specific to these particular cell types that we looked at. So then what we did is integrate attack-seek annotations, promoter annotations from EpiMap, and enhancer annotations in a matched cell type manner, and again compared those with what would be identified from bulk. And across the board, we see greater enrichment in the corresponding regulatory elements that match the cell type rather than what we would see in bulk. And so again, it's showing that we're getting a more fine-grained look into what might be going on at the genetic regulatory level at these particular cell types that would be missed if we looked at just bulk. So another thing that I thought was kind of neat, and so it's kind of, I don't know, I feel like I'm at odds with this. It's like it's an interesting finding, but at the same time, it might kind of pull away from driving, maybe it's like the enthusiasm of driving in this direction. And that cell type specificity is massively confounded by the depth of cells, right? And so here on the left, what we're showing are really just the number of E genes that we identified based on and how that tracks with cell type proportion, right? So as your cell types get more and more common, we're going to have more findings, right? And it's just because we're essentially having more observations that we get to collapse over. It's going to reduce the variance, increase the count, in our generalized linear model. And so previous works analyzing this data really sort of laid into that notion of like, look how many cell type specific things are being identified, and they exist. I don't want to make it seem like that's not, we had a whole slide covering that initially. But again, this is something that's massively confounded. And even in our data, if you just take a count based off what was significant and what cell type, we would conclude that like, yes, these eQTLs are massively cell type specific. But really, maybe what we should say is like cell type biased or use some other terminology, because if we relax how we consider sharing to maybe be shared by magnitude or shared by sign, we see a completely different story, right? And so if we look at just the effect direction, we see a massive concordance across the cell types, right? So it might not be the exact same effect, right? There'll still be heterogeneity there, but at least directionally, whether it's upregulated, downregulated, we're still seeing that. This might still be additionally confounded whether or not we're identifying more promoter-based eQTLs versus enhancer eQTLs. And so, again, I don't want to paint a picture that's saying like cell type specificity is not real. I think it is, but it's just a trickier sort of problem to work through than what, just a simple analysis. Yeah.
Do you think that there's a role for the sample size to confound some of these statements? So for example, if you do this analysis again when you have like 20, 30,000 or massive, whatever,
yeah.
you max out the EGs for all the cell types,
right? And
you redo the analysis. Do you think at that point you'll start seeing whether it's truly cell type specific versus biased, the way you put it?
Yeah, I mean, I definitely think a lot of that will get resolved through increased power, right? I think, just like trying to identify, how many GWAS loci there are, there's, you'll hit a saturation limit and there's going to be things that we'll never be able to say maybe with a lot of confidence, but I totally think so. I think it's that, and then also better phenotyping, right? Like we're just using the labels that the 1K folks said this is what we're calling the cell type, right? And whether or not it really is, these are all exactly the same cell type, right? Like we've had the conversation earlier about these discrete notions of populations versus continuous. So I think, to answer your question directly, yes, I think increasing sample size will help. At the same time, improved phenotyping and methods will help simultaneously, even within the same N.
So maybe I can think more on that. So when you say phenotyping here, you mean like the cell assignment to cell?
Yeah, exactly.
Pair that may vary across cell types and so on.
That's right.
So you showed lots of methods there. Are some methods more robust to some of those errors than others?
So as far as I can tell, not. I think it's still an open question, right? I think the field's just getting started and applying these tools to answer these questions, and so. What we did is basically apply MASH-R to all of our effect estimates from these tools. And so, I think that'd be a great opportunity for a method specifically to maybe better assess that particular question.
Thank you.
And then lastly, what we did was integrate our findings with GWAS to see do we do a better job about explaining heritability compared to what would be explained had we just done a bulk eQTL analysis. And so across the board, we do see that the eQTLs explain more heritability on average. And then that we do see, again, sort of common sense things popping up, right? If we were to look at monocyte percentage, we see exactly the monocyte eQTL annotations being significant, right? Or if we were to look at lymphocytes, given that lymphocytes represent sort of a collection across a lot of these white cell types, if we take the union, right, all the eQTLs identified across the cell types and just treat them as one annotation, right, we see a massive enrichment there. And so again, it just points to that leveraging these cell type specific findings can help better characterize like particular tissues or cell types and where to look next for disease. And so as just a nice example then, what we did to focus on our findings with rheumatoid arthritis. So at the top, we have a risk locus for rheumatoid arthritis. And then here, each sort of track represents the eQTL association statistics from our tool. And then this one here at the bottom is if you were to use our method with just bulk, like collapsing all the cell types together and ignoring it. And we really only see eQTL signals in these CD4 and CD8 cell types. There is some signal here in the bulk, but it's massively attenuated, so it'll be easily missed. And so while it's great that they kind of just align, it's not clear how this mechanistically might be related to another. And so we also integrated promoter capture high C that match these corresponding cell types, and we see there's contact between the promoter of this IL-6ST with this risk variant in both cell types. And then similarly, when we integrate markers for active transcription, we see peaks in the matching cell types, both around the promoter as well as where the risk region is. So this points to that this particular variant may be dysregulating IL-6ST in these two cell types, which then leads to rheumatoid arthritis risk downstream. I think there was some additional work where folks have followed this up with like some MPRA of knocking it out in a particular matching cell type and did see changes in gene expression at the level.
But you do see peaks in the other cell types. So here's just the
Yeah, exactly. So
the contact is cell type specific.
yeah, that's right. So I'm not sure if we have monocyte promoter capture high C to also see, because we, like you said, we do see this strong regulatory signals in monocytes, but it's completely null. And so that might, again, maybe be confounded by the amount of monocytes we have in our data, but I still feel like probably not likely. There should be a little bit more signal than that, but it's still, I think that just points to like how challenging this is, and it can be maybe easy to fool oneself by just overlapping this and looking at it. Really just, you need like a triangulation of a bunch of results. So this work has been pre-printed. I should say this is joint work with Stephen Gazal. We co-mentor Eleanor, who has just recently graduated and will be moving to Penn to postdoc with Bogdan and Breland Brown and Mike Gandel, so we're very excited for her. I know this was initially going to be a talk about JAX, but it's still there. So the tool is called eQTL. It's written in JAX. JAX is Google's machine learning library. It's like an auto-diff framework. It's really, I have a lot of fun with it. I've sort of tortured all the students in the group and said, like, it's going to be a steep learning curve, but once you learn it, it's worth it. And really, even if we're not using any auto-diff stuff, right, like the just-in-time compilation makes things fast, right? Even just on CPU, it's going to make it a lot faster than just pure Python code. So there's a benefit for that. And so just in conclusion, eQTL provides a very fast approach for large-scale single-cell eQTL mapping using count-based models. It's more powerful than the linear model. We have similar power in simulations compared to SageQTL, similar-ish power in real data. There's a number of reasons why they might differ. I'm happy to talk offline. Things were more enriched in cell-type-specific cis-regulatory elements compared to the bulk. And so acknowledgements, obviously both mine and Stephen's labs, Eleanor, Noah, who was a undergrad volunteer, Artem, as well as folks who've commented on provided feedback on the project over the time. Happy to take any questions. Thanks. Oh, and thank our funders. So.